Okay, so this is a retape of sex-linked traits or sex linkage. So I'm doing it by myself. I don't have anyone in the class, and that's why I'm not wearing a mask. Okay, so Emily, this is for you as well. All right, so on page 7 of the worksheet, we've already dealt with sex determination. What's the chance of having a boy or girl with each conception? And we worked out that it's 50% chance each time. That was in the last lesson. So today we're looking at sex-linked traits or sex linkage. So now you've got to be very careful because we've dealt with 3 different types of crosses so far. So let me just go back so we can see the progression. All right, so number 1, we dealt with normal, recessive, autosomal crosses. Okay, so for example, two heterozygote parents, even though, for example, they do not have albinism, they are able to, 25% of the time, they could have a child who has albinism. All right? Then, number 2, we dealt with dominant autosomal genes. Okay? So now just remember that autosomal refers to the fact that these genes are found on the non-sex chromosome. So in humans, it's from pair number 1 to pair number 22. Okay, so recessive or autosomal recessive crosses, both parents can be carriers, but they can have a child who has albinism. Autosomal dominant, and the example there was polydactyly, or in your, because this is not in the textbook, if you look on the notes, in the notes on page number 6, we talked about the disorder called Huntington's disorder or Huntington's chorea. Okay, so I'm just going to abbreviate it here HD, you can abbreviate it HC, but I think the newer term is Huntington's disorder. All right, so there, if the parent has one dominant gene, the dominant gene is the mutated one, and that parent marries, okay, so a male marries a female, and he has Huntington's disorder. Okay, but the problem with this is that this gene, even though it's present, they usually only get symptoms much later on in their lives, so in their late 30s, 40s, 50s, and by then they might have already had their children. So if we did a Punnett square over here, all right, so the gametes from the male parents, I'm just saying that's the male, would be big H or H, and then from the normal parent, the normal female who does not carry or have Huntington's disorder, her gametes are the same. All right, so you can see here now that there is a 50% chance that the children will have Huntington's disorder. And once again, it will only symptoms only usually show up much later on in life. So you need to be careful here, all right, because autosomal recessive, the person needs 2 recessive genes to have the disorder, like albinism, or think of pea plants, tall plants crossed with short, wait, let me just do that again. 2 tall plants that are both carriers, once again there's a 25% chance that the next generation of plants are short. So go and have a look at that again. We did a lot of those examples. Okay, so now we're looking at the 3rd type of cross. So this is known as sex linkage or sex-linked crosses. So now we are talking about genes that are found on the sex chromosomes or the gonosomes. So we're no longer talking about pair, in humans, we're no longer referring to pair 1 to 22 of the chromosomes. We are now referring to the sex chromosomes. Okay, so if you think of that was female, I just wrote male and female. If you think of, it's my little cloth. In males, in human males and in many other mammals, they have XY chromosome. And females have 2 X chromosomes. Now, in reality, the X chromosome is longer than the Y, and in females, they have 2 long X chromosomes. So if you think back now, we always talk about homologous pairs of chromosomes. So for females, they can have 2 alleles for each of the genes found sex chromosomes. So they could be heterozygous, homozygous dominant, and homozygous recessive. But now when you read a problem that refers to sex linkage, they're going to say that. So here, you're always going to write X, and then you're going to use superscripts to indicate the genes. So a female can be normal in terms of color blindness and hemophilia. We'll get to that now. So they can be normal. Okay, so let's do hemophilia. Hemophilia, the blood clotting disorder where they don't have a certain enzyme and therefore their blood doesn't clot and the platelets don't shoot out those fibrin threads which trap the red blood cells, which you might remember we did in grade 9. Okay, so for a female to have normal blood clotting, in other words, not to be hemophilia, they often use the letters big H to represent normal. So she could have 2 normal alleles, or she could be a carrier, all right? So she's either homozygous dominant, or she's a heterozygous for blood clotting, which means that she can clot her blood, all right? But then a female who has got hemophilia, all right, she would need to have 2 recessive alleles, all right? So it's pretty much, it's the same as when we're talking about recessive autosomal crosses, but we're now using the letters X and Y to indicate that we're referring to the sex chromosomes. All right, so that's for a female. Okay, now in terms of the male, he's only got 1 allele. He doesn't have another one. That little Y chromosome is too short. So they only have 1 allele, okay? So a normal male who does not have hemophilia, we write X, Y, and he would be big H. He doesn't have hemophilia. But a male who does have hemophilia would be X, little hy. In other words, males cannot be carriers because they only have 1 allele. All right, and it's the same for hemophilia, color blindness, and there are other disorders. And whatever they give you, whatever you see in the question paper, don't let the name of the disorder confuse you if it's something that you haven't read and heard about before, or that we haven't dealt with in class before. You look for that word sex linkage, or that it's a gene found on one of the sex chromosomes, and then you immediately know that you need to use this format of writing down the genotypes. You have to write down XX for females with superscripts for the actual genes that are present, and the same for males, but you never write anything above the Y because it's a much shorter chromosome. There is no allele. In fact, they sometimes, and you don't need to know this, but they say that males are hemizygous for genes that are found on the longer arm of the X chromosome because they only have 1 allele, whereas females can be homozygous dominant, heterozygous, or homozygous recessive, and then have that particular disorder. All right, so I'm going to stop now because I want you, let's just see here, on page number, what's it now, 7. Okay, let's do the first one. So we're going to do the first cross, and then I want you to try up to number 5 by yourself. So we're going to do number 1 on the worksheet on page number 7. Okay, so there it is. So it says, and you've got a little summary there now. I've actually, there it is. There's the little summary in the block, and you can see that it's exactly what we've done on the board now. So number 1 says, a woman who is heterozygous for hemophilia. Now, because this is in the section called sex-linked traits, in the actual question it doesn't say it's sex linkage. But if you get a question in a paper, November or wherever prelims, it will tell you this is a sex-linked disorder. Okay, but it doesn't say it in the wording of the actual question here. All right, so assume it does. A woman who is heterozygous, so remember she's got 2 X chromosomes for hemophilia. So she's got a normal gene and a hemophilia gene, and she marries a normal male. All right, so the Y chromosome does not have a gene for blood clotting, and he is normal. His blood clots normally. All right, so once again, okay, that is P1. Then we can show meiosis, and we can can show the gametes. So half the female's gametes will have the normal gene, and half of them will have the recessive gene. In the case of the male, half the gametes will be carry or have X chromosomes with the normal gene, and the other half of the gametes will have a Y, no gene for blood clotting. All right, so if we show fertilization using a Punnett square, all right, so X big H, X little h, X big H, and Y. So X big H, X big H, little h, X big H, Y. X, okay, let me just check here now, little h, Y. Okay, so the question said, what are the possible phenotypes of their children? So we can see here, F1, the genotypes, and you would write it out. So it's 1X big H is to, I'm going to do the females first, 1X big H big little h is to, I don't want to go off the board, 1X big HY is to 1X little hy. Okay, we're still on the board. So they just ask for the phenotypes. So we can see phenotypically, all right, that there would be one normal or not a female who's not affected by hemophilia. All right, try and use the word hemophilia. One carrier female, so she also doesn't have hemophilia, but she could pass it on to her children, okay? And then here, one normal male who does not have hemophilia, and then one hemophiliac male, because he's got, well, there's a chance, okay? So with every conception, there's a 25% chance of having a normal female, a carrier female, a normal male, or a hemophiliac male. All right, it doesn't mean they're going to have one of each type of child. Now, what you've got to now look out for, okay, is how they ask questions. So let's just practice with this one. So what is the chance of them having children with hemophilia? Only 25%. Children. So I'm looking at all 4 blocks. Okay, so let's just write it here. Children, with hemophilia. What's the chance? It's only 25%. So X little hy. What is the chance of them having carriers of children who are carriers of hemophilia? Well, that can only happen in females, and it's also 25%. X big H small h. What is the chance of them having a son with hemophilia? Now it changes. Okay, there's a 50%, well, there's a 50% chance with every conception that it will be a boy. And then if you look at those boys, okay, there's a 50% chance, that there, let me just move this over a little bit. Okay, so hemophiliac child is there. Carriers of hemophilia, 1 block. Son with hemophilia, so 50% of the time they expect to have boys. You know, that's the possibility of conceiving. And then half of the boys, so 50% chance of having a son with hemophilia. Okay? So what if we said, what is the chance of them having a daughter with hemophilia? Well, no, none. But they could have a daughter who's a carrier. Okay, so half their daughters could be carriers. Every conception, there's 50% chance of having a daughter who's a carrier, or a 50% chance of having a daughter who is normal. So you've got to read what they say. Are they referring to children? Are they referring to sons? Are they referring to daughters? So this one on page 7, number 1, was pretty straightforward. They just wanted to know the genotypes. Okay, so that's like an introductory problem. So then I want you to try number 2, 3, 4, and 5. Now on Monday we marked up until 4, and we went over the test today in class, Wednesday, and tomorrow we're going to go over number 5 and then carry on with all the questions on page 8 and 9 and 10. Okay, so I'm going to stop here now. You're going to try 2, 3, 4, 5, and then I'm going to make a video of that as well.